So ultimately, we were at this point where we said, if we have done the statistics right, we have seen that there is a signal, a statistical signal, that that there are some differences between, for instance, residues inside proteins that bind inside proteins and residues that bind between proteins, and we have developed a method that predicts it. So we have clearly established that statistical is more than statistical significance. We can actually do much more. We can predict, right? This is more significant than an e-value. Now the question is, do we understand? And that gets us back to this issue of black magic. And this issue of black magic, to cut it short, there's this misunderstanding that this is black magic. And the misunderstanding is caused, black magic, let's call it black box. This misunderstanding is caused ultimately by the lack of careful testing. So one problem really is the degree to which the generalization ability is not tested. It's overtrained. So most publications in machine learning really overtrain the device, and then you get sort of a bad reputation for the field. The other issue that comes on with a black box, the idea of a black box is that the knowledge, the prediction happens in these green connections, right? And you cannot tease out from these green connections what they have learned, right? That's wrong. You can. And that's the next big misunderstanding. So there's a variety of ways to really tease out from these connections what the system has learned. You can do that by seeing how it reacts to simple predictions. You can do that by sort of removing some of the connections. You can see by really testing what in the input vector causes what green line to be more on. And then you can sort of build up a signal that says, well, conservation at position 5 is a very, very strong signal, right? The problem with this, with doing this, and the reason why people have begun to do that about 20 years ago, when machine learning was introduced into molecular biology, or 25 years ago, and then people really opened the black box. The problem is what they found. What they found were these rules that if there is a glycine 3 positions away from a proline, and if there is an arginine somewhere close to the beginning of the window, and if there is two lysines here, and if they have a conservation rate of 25, and. So you build up a rule, and an expert listens to this rule and says, after the third if, they switch off. So expert knowledge goes to the point of saying there's a proline. Proline is a special residue. You want to tell me that proline is a special residue? I know that. Okay. Proline protein binding, most likely not. So if you have this F here, the tyrosine, that's more likely. The threonine. So other residues are more likely to bind, but the proline not. So that rule I don't believe. If the rule is simpler and it says a tryptophan would bind, then people would say, okay, I recognize that rule, and I believe that the rules you extracted make sense. The problem is, ultimately, machine learning is applied to situations that are so complex that the rules you extract are complex too. So if we used these simple rules of what people know, the proline breaks a helix, or the tryptophan is involved in protein binding, these simple rules alone give a very, very poor prediction. In fact, those simple rules alone give a prediction that is worse than random. Okay? Yes, this is expert knowledge, but it's actually not really standing up to the latest data set. What we apply it for is when we don't have these simple rules, okay? And then when we tease out the simple rules, they don't exist. Because by definition, we apply machine learning exactly when simple rules don't cut it. And that's the big misunderstanding. The big misunderstanding that the box is a black box because we cannot understand what's happening in it. We can, but it's not the kind of understanding that we are used to, because the problem is too complex. And I believe this essentially is one of the big misunderstandings in the field. Ultimately, machine learning, the solution of machine learning is as complex as the problem, right? And that typically implies Again, there are exceptions, but that typically implies that you can't understand these rules. I assume that if we sort of teased out how the deep learning is doing chess, then most likely you would get back to simple rules, to simple statistics for the first few moves. But I haven't done that yet. Now, when the first statement here was that in order to really distinguish between sort of understanding and prediction, although it is somehow similar, the task is we have to be able to say that the system generalizes. It predicts not only for my data set today, but also for my data set tomorrow. And for protein-protein interaction, to establish that is more challenging than for other things already. Okay, let's get back to the way we define protein interactions. So this external interactions is only the red ones here, the two external interactions. And essentially what we do is we try to predict the interaction of AB through some sequence-derived features. For instance, from this server predict protein. And then the simplest idea to predict protein-protein interactions was the one I briefly may have mentioned this before, is that I have one protein here, a window of 1, 2, 3 adjacent residues here from one protein, and 3 adjacent residues from another protein in green. And essentially, I try to predict whether or not the central in this window, the central 3, depending on how you set it up, are by some definition in contact. Less than 6 angstrom, or whatever my definition is, right? And this system alone completely failed to do it. So I tried to do this with a neural network initially many years ago. And my rationalization is this completely failed because a lot of what I see in my in silico, so in order to create these windows here, I take a protein sequence and I cut it into bits, right? And I shift this window through, just like for secondary structure prediction. And the same is true here. Now, what will happen is that many regions will, in the computer, meet each other, that in nature will never meet each other. Why will they not meet each other? Why could they meet each other somehow in the computer and not in reality?
If I take any part of the sequence, it could be just inside the protein. So
it could be inside. So that's the first issue. So the simplest way of saying it, half of the protein is inside. So half of these windows will somehow, or at least a quarter of these windows, no, 3/4 of these windows will have one fragment that is inside. So we'll never be able in nature to see because the protein binds. If we talk about transient protein-protein interaction, we're talking about two folded objects that bind only on the surface. They can unfold a little bit, but not completely. Okay? So many of these regions that I can match in a computer will not match in reality. Another reason could be, what do you think about other reasons why these regions may not meet in a cell?
Maybe they are different localizations.
Yes, so these two proteins never meet because one is nuclear and one is secreted, right? So if we put them into the tube, if I put them into the computer, maybe they would actually bind. But all the experiments that I have are from real life proteins and I don't see the binding, right? So I see them as wrong. This is another reason. They may not even be expressed, so they may be from different cell types. So in one cell type, this protein may not be expressed. So it would bind the green one if they really would be expressed at the same time or in the same cell type, but they are not. Okay? So there's a lot of reasons why this is likely to trip over, let's call it an ocean of false positives, of things that in nature will never meet. Now, you can try in a computer to sort of filter this out. You can try to sort of predict surface residues and then only have surface binding. You can sort of, the complement of that, predict residues in external interfaces. That is what we talked about before. So we essentially focus on the parts that really are known to bind other proteins and say those will clearly be in the windows. You can predict residues that are saturated internally to simply say those I will clearly not use, whatever binds internally. You can predict subcellular localization and say that nuclear and secreted proteins I will not bind here. You can predict residues in substrate interfaces. So if the protein binds DNA, it will not bind at the same place another protein, or unlikely, or bind some other smaller ligands, enzymatic activity ligands, smaller things. Again, that will interfere with protein-protein binding. Then we need sort of a better definition of domains, because protein bind by domains. And then essentially, after all of this has been done, we can predict protein-protein interactions. Okay, the first time I tried this neural network for protein-protein interaction prediction was 1987 actually. And putting all of this in place took 10 people's 10 years. Or 10 people, roughly 10 years. So this is a large research project. And at some point, Tobias, that really happened in Munich here, Tobias put it all together in some sense and predicted protein-protein interaction. And again, one step on that way that Tobias sort of contributed to with Tatiana was the prediction of subcellular localization that I talked about. Now, let's get back into this challenge issue. The challenge, let's begin with the first issue. The first issue is about redundancy reduction. Redundancy reduction means, so there is some similarity here for all the proteins in this area. And typically the way we do that, we address that in cross-validation or training and training testing split. We take one representative from each family. Okay? And by taking one from each family, we reduce the redundancy. That's very well defined. It's a lot of work. But for the protein-protein interaction, we actually do not have these clusters easily. Because what we actually have to cluster is pairs. And clustering pairs is already not so trivial, because, let me give you a simple example. Say, we had a world in which we had markers. That world exists only in dreams. No. So say we had 3 proteins, A, B, and C. And they were defined such that no pair of them is sequenced similar to each other. So they're all very different proteins, okay? Now, when A binds B and A binds C, since B and C is the different protein, you could argue that the binding between AB and AC could be in different clusters. Because C is different, and B and C is different. And that could very well be true, but it could also be true that if you look at A, so this is A, and it binds B here, so this would be A, and this would be B, and it might be true that C is just binding to the same position. B and C are different proteins, but the residues at the interface to A might be similar. The structure at the interface might be similar. This may exactly be why 2 different proteins can bind at the same position to A, right? And if you have a case like that, then you may begin to argue, okay, in the case in which the physical reality of binding between A and B and A and C is the same, they should be in the same cluster. Right? So this now gets complicated, because it throws up another set of definitions. When do you call this a similar binding site? And what I didn't talk about is when I look experimentally, I have protein A and B and they bind. Okay? And I know, let's just say they bind this way. Okay? Now the question is, if 10 people measure this, 10 different experiments, will I always see exactly the same binding? Or will I sometimes see that they bind this way, or may even be more different, right? And it turns out that when we looked at different ways of doing the same experiment, high resolution structures, in many cases they differ. And now how to specify that is a little bit tricky, but there's a lot of difference between them, and this lot of difference goes to examples where they really bind completely differently. Okay, why is this the case? Well, was another surprising result that we had. We actually don't quite know. This is a very definition of a surprising result means you don't understand what it means, right? But it's an observation, and that in turn means since there is sort of sometimes you see the same, sometimes you see slight differences, and sometimes you see large differences. So when do you cluster it together? And essentially, we cannot make the decision. This is, by the way, why we got into this story, simply for the clustering bit. And we decided that the only way to do it is just stay away from it completely. So we still have an issue. Now, the next issue that we have is so that's essentially what I talked about. Let's not have the same pair in training and testing set for any of the two. But now comes the next issue, and that's the story that goes back to Marcotte and Park. So what they looked at methods that predict protein interaction pairs from sequence, just what I'm talking about. And what they discovered is that methods behave different or perform differently according to 3 different cases. In case 1, the protein pair AB has been used for training, but either A or B, not the binding. So what they say in case A is that both, so I want to predict that A and B bind, so this is my test case here. Super strong, the green. So this is AB is the test interaction. And in case 1, both A and B have been used for training, but not as a pair. So the training set never contained the pair AB. It contained AC, it contained BD, and other things. Okay. In case 2, either of those 2 has been used for training. And in case 3, none of the 2 has been used for training. Okay. And what they observed is that the performance of methods decreased. So when none of the 2 had been used for training, in fact, the methods performed much less well. Okay? I'm not going to ask you to explain this, because this, in principle, was unexpected. It's another definition of surprise, so we can come up with a lot of explanations, but actually it was a surprise.
But
now, yes, sir?
Why was it a surprise? When you get less information on A and B, the further you go down in the case.
But so the point really is that, yes, so if you imagine that we have cases where really the binding site is the same, but what we actually have here are methods that look at the entire protein A and the entire protein B, and they predict the binding. So they're actually not seeing binding sites. So they will actually not see the difference between A binding B and A binding C in the same binding site, when B and C are different, or A binding B and A binding C in different binding sites. For those methods that will look the same. Okay? So they just grind up, all of these methods here grind up the entire sequence information. And then that means that ultimately at least 1/2 of the binding partner, in terms of looking at the entire sequence information, is as different as in any other case. Right? But you may argue that you expected it. Fair enough.
Less information still. Maybe it's not coded explicitly.
But if you really learn what it is that makes proteins bind. So if your method learns A is a binder, and you don't put A into your training set, then it cannot even learn that A is a binder. And then in that sense, you're right. You have less information. But if you trust that your method extracts anything more reasonable than A is a binder, the story gets more tricky. And none of the developers of these methods is really ready to say that all they ever learned is A is a binder. Okay, so here are 2 methods, pipe 2, sig-prod, both sort of predict protein-protein interactions pairs. from sequence alone, and the C1, so both have been used in training, A and B. This is the performance in red, and here's C3. So C3 throughout the area of recall here, essentially throughout, for SIGPROT completely throughout, the green is always below the red, but here it's sort of largely below the red. Clearly it's below the red in the region that matters most here, where we have high accuracy, here high accuracy and low recall. Now the next question that we have is, all of this clustering has been done on the positives. So remember that the way we are going to play this, the way these methods are developed, is we have a set of positive interactions. A binds B. We have an entire organism, human. So all of these are human, or one particular organism. And we say the negatives are, so this is experimental. The negatives are the, let's call it negatom. So some that we have that are experimentally measured to be binding and not observed to bind. This is the one that I'm talking about. Not observed to bind is, I simply take human, all 20,000 again 20,000, whatever has not been observed is binding, or there is some refinement that I talked about. This is a high reliability experiment, and then there's a low reliability experiment of A prime, B prime, and that is not used as a positive training set, but it is used to exclude not observed. It's not clear what I said. Okay. We have very reliable information about protein binding. That reliable information is the only thing that is used as positive instances of binding. Then we have low reliable experimental information. Somebody has some indication that these two proteins bind. That's not good enough to positively train that A and B bind. But it is good enough to say, I'm not in reversing that. Anything that has not been observed is reversing the observation in some sense. The low resolution experimental information is good enough to say it will not count as a negative. Okay? So essentially those low reliability cases I will exclude entirely from my data set. Okay, but now back to the question on the whiteboard. So by negating essentially the positives, so by using everything that could bind and that has not been observed as negatives, I have an artificially created data set because I have not observed it. Okay? Again, there are some negatives here that are experimentally observed, but it's a tiny fraction. The negatom is a tiny fraction of the positives. Okay? So we do whatever I talked about so far, the clustering of the protein interactions. A binds B, A binds C. Can I use A and B? The class 1, 2, 3 that I talked about is all for the positives. Now the question is, should I also consider sequence overlap or redundancy reduction for the negatives? It's a very simple question, right? Yes or no? Begin with a binary answer. Either way you fall, you have to argue for it anyway. So it's the same number of words. Who wants to go forward? Nobody. Are you all ready to say yes or no? So yes, so no. So yes essentially means I have to cluster also the negatives. Again, mostly in some sense you would call it simulated data. It's not simulated, but it's the negation of the observed data, right? So yeah?
Just one question. Don't we make like a lot of assumptions if we say just because we didn't see it, they don't bind?
Absolutely. We absolutely make a lot of assumptions. And this is, you may have been there on the whiteboard talk when we talked about inferring the connections between you guys. And you absolutely know that students that this reporter has not seen, if you remember what I referred to infer those makes no sense. You just have been sick, right? So, but as in a statistical set, that's the only reasonable way of doing it. Put it differently, for machine learning, we need negatives. The negatome data set is tiny, not even a tenth of that. We do know that most proteins do not interact. So if you couple 90% positives to 10% negatives, you get exactly the opposite of what you want. You assume that every protein, if you ask for 20,000 proteins in human, how many do I interact with? Most likely, the number of negatives to the negative to positive ratio is at least ballpark 100, say. So by making 1/10, you go 3 orders of magnitude in the wrong direction, right? So while you are right that we make a lot of assumptions here, and in fact it's even worse. Let me put another step on it, Sandra. The fact is you develop the prediction methods to find interactions that nobody has seen before, and exactly the set that you train to be negatives. So you actually want a device. You want sort of a student who tells you as a teacher you are wrong, right? But you still hope that. So to begin with, you need it, all right? So there is if you want to do machine learning, we could try it with these. There's a class of machine learning devices that only learns from positive evidence. But in that context, this is yet another step of. We are trying this for another story that I talk about next week, which is the effect of sequence variation. Dimitri, in fact, will be trying that, has been trying that, is trying that, and there is already sort of not quite working. But here, the story is more complicated. So we need negatives, and then we need to make some assumptions. Yes.
I just wanted to ask if we can expect the negatome data set to be just as reliable as a positive one.
No. Absolutely not. And the reason is simply because the way it's done, when you see an interaction, you see it. When you don't see an interaction, it can always be that you did not find the right conditions for the interaction. So no matter how much you sample, you can never sample all conditions. While seeing it once is enough to make the point. Or seeing that it happens in a cell. So in that sense, the negatives can never be anywhere near as clean as even the experimental negatives not. And in fact, that is one of the reasons why people tend to not put negatives into databases, not into experiments, not into papers. You, Matthias, you had this story with experimental negatives, right? You came up with this. No, I can't remember. On Tuesday? Tuesday is too long ago. Totally understand. Yes, we make a lot of assumptions. But this was a sort of mean detour to get me off the boat, right? So the question really is, for the negatives, that again, a lot of assumptions, maybe even more are wrong than right. Now, if more are wrong than right, then the story will not work. So I believe ultimately in this rationale, we hope that the machine learning device will find a signal. It could still be that more of them are wrong than right, as long as there's a consistent signal in the negative set that sets this negative set apart. And most likely, in doing this here, way more are right than wrong. Because simply most pairs don't exist, right? So it's most likely a factor of 100. So yes, we make many mistakes, but at least 100-fold we are right. Simply by the force of numbers, not by the intelligence, right? So bringing back the question, should we now sequence redundancy reduce? You're getting me today. So sequence redundancy reduce the negatives. Yes or no? Nobody argues, so vote. Who's for yes? 1, 2, 3, 4, 5. And if I wait long enough, it will be everybody right. And for the yes? That was the yes. Sorry. Can we also have 5 for the no, so that I don't have to write? So who's for the no? 1, 2, 3. Ah, no, 4. Well, it is right. Split vote. I don't know. So nobody has ever done that before. So when the student came and said that has to be done, the teacher said, "You are an idiot." So the expectation of those with more experience were completely other than the unsplit group. But the fact is, so nobody has ever considered it, and therefore it was considered not relevant, but it's wrong. So the majority in the room, the 5, were more right than the 4. So let me not get into too much detail, but considering it or not considering is the difference between the two is the delta here. The blue is, there's a complicated point. The plus delta means that all of those methods above this line overestimate performance simply because they did not filter the negatives. That's ultimately the message here. Conclusion, we have to consider negatives. We have to consider them in terms of redundancy reduction. Now that gets us to a point where now just drawing the idea of redundancy reduction becomes something that is no longer trivial. So now I would have, so I can spend the next 10 minutes on the next 2 slides and go from this redundant networks to this non-redundant network. So you will understand what you have to do. You will understand how you have to do it. But you see, already the simple idea of redundancy reduction suddenly gets complicated. The next challenge is the data issue. So let's give you one example. What we know is we do not have enough experimental data. Okay? And somehow we already touched this issue. Since we do not have enough experimental data, if we have A binds B and A prime binds B prime, where A prime is similar to A and B prime is similar to B, then since we don't have enough data, maybe we should use this sort of non-redundant, at least for training, because we don't have enough data. Clearly, if we have A binds B, my initial example, and A binds C, and B and C are different in terms of sequence, then we should use both. That's what we had initially assumed, and that in fact is wrong. But I forgot another story. The first question really is, how much data is needed? How can you find that out? How can you cannot find out how much data you need. So what you can find out, if you have enough data, you solve the problem. If you have enough data, you have captured the important input vector, the important input information, you can clearly predict. Clearly predict is without any error. That does not need 100%, okay? You can get it to the ceiling. The ceiling is defined as experimental mistake, or ceiling is defined as mistake in nature. Sometimes proteins misbind, or whatever your ceiling is. But if you reach that ceiling, then clearly you have enough data. That's clear. But if you don't reach the ceiling, how do you know how much data more you would need? How could you estimate that? We've briefly discussed it in one of the,
I think you need to basically take a look at how many parameters you want to find, and then I think 10 times more, 100 times more.
So this is one way of doing it. In fact, that's a very important one. But it's a rule of thumb. So the rule of thumb is essentially you need, I don't even need to write it down because it's a rule of thumb, right? So you need 10 times more samples than free parameters. And you immediately see with the rule of thumb, this is I can easily show you how this is nonsense. Say my training example has a pair of A binds B, and A binds B prime, and B prime are 99% sequence identical. Then I would have two samples, but they almost contain no additional information, because it's essentially the same thing. So now the issue becomes, I have to measure actually the overlap between the samples. The overlap I can not quantify. If I could quantify it, I could classify my problem. Ultimately, I need to run the machine learning in order to get a measure for similarity. Machine learning evolves a measure for similarity of these input vectors, right? And that defines, ultimately at the end of the day, that defines what is overlap. The overlap that we discussed so far about our redundancy reduction always was not overlap in terms of the vectors, the input vectors to machine learning. I was an overlap in terms of what other method could I use? I could use homology-based inference. In that sense, it was redundancy reduced, right? So with respect to the vectors, the story gets more complicated. So this is a rule of thumb. The rule of thumb, in many cases, may not hold. But it gives you an idea that you may need more. But there's another way of doing it. What else could you look at? How else could you sort of get some idea how far away from the number of samples you are? Do you have any idea what you could do?
Can you look at the previous work?
Yes, so absolutely fair, sir. It always is a good way to start and see what Google tells you, what previous work is, what they say. But assume that you really are at the cutting edge, that you do something that nobody has done before, and you cannot look at previous work. So, because your input, so you can look at the rule of thumb 10 to 1, but you do not know what the overlap for your samples is. You predict, you have the first method that predicts protein-protein interactions from sequence alone, for instance. What could you try? Yes.
We could take part away from the training data and see how much worse it performs.
Yes.
And from there.
So the most extreme one, or the extreme one here on the whiteboard, simply try half the data. I know, or some hit it substantially. Half is a relatively substantial hit, right? And see how much does the performance go down. Okay. Now say I took half of the data away. So say I fixed the recall. So in my accuracy for a fixed recall for the full data were, say, 35%, and my accuracy for the half would be 30%. So I can clearly see that half the data is not good. And say my random in this particular case, because protein-protein interaction ultimately is something in the ballpark of 0. That's random guesses. You have to find 1 in 1,000, that's 1 in 20,000, and it's a pretty small number. So by random, okay, I'm missing the error estimate here. Say by the error estimate, this is an absolutely significant difference. So what you need, what you learn from that is that halving the data makes a big difference. My question to you now is, how much data do you need?
I just have one question, because let's say, for example, if we added double, like 2 times f, for example, nobody tells us that the improvement will grow like by, I don't know, 10% or 5%. It would probably be completely, like, just improve it by 1%, so we don't really know how much we have to add.
I dislike the use of word. Probably you don't know. There's only one word that I want to correct in what you said. You're right 100%, 99%, oh, I didn't count the words. You're right except for the word probably. What 2f will look like, you cannot say anything about probably. Okay, you have no evidence here for what is probable for 2f, and that essentially is anyway your point. You cannot estimate, but That's the challenge, right, in science. The challenge is to get to a point with all the material that you have to get to some guess. What would you do if you saw that?
I could make an upper bound at least.
Would you do that, Matthias, or would you do something else? Yeah?
We could check if the accuracy is already converging towards the 35 or if it's still growing.
Yes, how would you...
Keep on adding data points between half the data set and the full data set. If we get a trend that is going upwards, then we can try more. And if we already see that 3/4 of the data set is at 35, then we probably won't.
This is one thing you would do. I would do, Johannes, is a good idea, but I would do something first, before I do that. Yes, I would add data points. But the first thing I would, so looking at these two numbers, you're absolutely right, Sandra, it cannot is expected to be linear. But they, given random being, let me put my random here, 0, giving random being 0, it's actually surprising that it doesn't drop more, right? So I would hit the system immediately with 1/4 of f. So I use 1/4 of the data set. What's going to happen then? And if that is not enough, I'm going to go smaller. I want to see that the system cracks. And again, we have an example now where we are reducing this data set here, and we're reducing this data set, and we are getting to a point that is clearly above random. And it seems that the system extracted some very, very trivial information. So we have to somehow redefine random. So there is some relatively trivial information in the input vector, and all it does is it extracts this trivial thing and needs very, very few data points for that. Where again, this is an example where you open the black box, where you see that essentially what happens, so in this example that we have, to just stay in the language here, we get, for 1/10 of the data set, we get to 28 or something like that. You see that most of the data points add relatively little. Okay, so if we were to generalize from this, so 90% of the data move it to by 7 percentage points, so doubling it maybe is 42 or something like that. Okay, and then again, now you come to the point that we may have a function that we have to fit. Gary's, anyway, so the 3/4 point, Johannes, is a right one, but in a case like this, since I would be surprised that the 30 and the 35 are both so far away from 0, then I would begin to hit it in the other way. And that again is not really opening the black box, but it is part of understanding something about your data. Okay, and while you're right, Sandra, that we are not really in a position to gauge and to judge and to predict what will happen to A2F, we are beginning to see, in this particular case, we saw that in fact most of the doubling of the data will not improve much because it is very difficult. What we still don't know is whether we're going to a ceiling. So is that 10 times f is going to be at 80? Or are we at a ceiling where this is somehow 35 and the ceiling is 38? And that functional shape we cannot really see. We can try to see it, as Johannes says, by filling in some points in between, hoping that I get a curve. But this curve is always assuming that somehow the curve continues and that there is nothing special for the data that I have before. Or to say it in a different way, for the data that we have in protein-protein interaction prediction, we believe we are so far away that fitting a curve is unreasonable. This is our impression. So what we clearly know is more data helps. So again, we have a difference here in the precision between using more data. So there's a realm in which clearly more data helps. And then there is the issue that not all good data is really relevant. So for C3, putting in, that is ultimately the story, if I have AC and AB, I can only use one of them, otherwise I heard C3. In particular for the class C3, so I've neither used A nor B for training. So we are in what you call I have little information about the pair. When we are in that class, then we particularly heard it by saying, by putting A as a multiple binder into the story. Then this network, because it has so little information, as you correctly pointed out, will jump into simply saying A is a multi-binder. By presenting AB and AC, although B and C are different, essentially the system goes into only learning A binds always. Ultimately is the point. And that's the problem. For C3 it doesn't work. For C1 and 2 it's a slightly different story. But ultimately what it implies is that when you do protein-protein interaction prediction, the problem of cross-validation becomes a very complex one. I call it's a squared problem. In fact, it means that there are very few people who can actually create clean data sets. And most extremely put, there's one in the world at the moment, and that's the face of it, of that single person. Extremely tricky. He did one data set. It cost him a long time. And now the issue is we have many different organisms, and we have new data, and we have to build a new data set. And so several students were freaked out when they had to sort of repeat the complexity of this. Highly non-trivial. Okay. Now the third issue is we want to predict that A binds B. What's the method? So this typically for people who apply machine learning is what they spend most of the time on. Typically this is the part that in molecular biology, this challenge 3 typically is at most 10%. Most of the challenge goes into understanding the data, creating the data, cleaning up the data. Once we have the data, the challenge 3 is relatively trivial for computational biology. So, what methods to use? Any suggestions? So I want to predict that A binds B. We have two proteins A and B, and all we want to predict is they bind. Not where, but that. So I essentially compare all proteins in human. I want to find the pairs that do bind.
You're talking about network architecture right now.
Yes, well, not quite. So network architecture is sort of one answer. I'm going to use a network. And I assume with network you mean a neural network.
Yes.
Yes. So that would be the question. Do you use a neural network? And if your answer would be I use a neural network, then the question would be about architecture. Yes.
I would use a network.
Okay, so Matthias, how would you use a neural network?
Maybe a Siamese network, or how it's called. So you take one network, let's say fully convolutional, and throw it at both proteins. Then you get an output vector, let's say 30 units, and like another network on top of that with a dissimilarity function.
Yeah, good work. It didn't work in our hands. But we tried. We tried in two ways. So we tried it in the way that I initially showed, where I had this sort of old-fashioned kind of idea where we simply cut out a part where I don't necessarily have a convolutionary model of the whole protein sequence, but I just have a part. We threw that at a normal neural network, we threw that at a convolutional neural network. We tried exactly the idea where we had the entire sequences, throwing it in, with an autoencoder. So far we have not made them work. But we're trying. We are still working on your idea. I mean, still working. Something else came in the way, but we will continue on that idea.
Can you share the data?
Yeah, sure. Let's talk about that later. So that one didn't work. Another idea. Or the solution that we have, at least in our group, that works. And ultimately the problem with that is it zoomed into one particular class of proteins. Essentially it can predict for one particular class and not for any other. And for that particular class, it's not as good as experts for that particular class. But anyway, so another idea. So ultimately, really the question is about what type of machine learning device. How do you do it? What algorithm, so to speak? And again, we believe that the answer will be deep learning. Yes, or will be something like that, but I cannot show you the proof. I can only show you how we fail. We brilliantly fail to do it. Any other idea? So, think about it. You form groups. Can we do pairs or triplets? So, two triplets and a quadruplet. So this is one solution ultimately where we look at the pair of the windows, but we pre-select the windows such that we select only pairs of windows where we have hotspots, and then we mark the hotspot prediction into those. And this is something that in this particular way we have not tried. And that ultimately is true because the way we have tried took too long. And the student who did it didn't come to it, and then other projects. The combination of these two ideas, I believe, is something that we should do. So this is something that remains to be done. So we haven't tried that. Any other idea?
Well, we were talking about, well, if our data set is not large enough, then maybe the neural network is not motivated, so it will easily overfit or do something like that, and then you could use other models
such as
random forest or decision trees.
What is the input information for that?
Sorry?
So what would you take as input then? If you took a random forest, so how do you code the input of two proteins? You were saying I have an idea, or you?
No.
Yeah, I'm not sure. I feel unsure about the sort of the topology of.
You still seem to be more sure than others in the room.
But no, I wouldn't. I don't have a good answer to that question right now.
Anybody? Any idea? How could you sit here?
Well, good.
Yes. And ultimately, you have the full complexity of the solution on the slide. And ultimately, I will even leave it here, meaning that I will not really get into the details of the algorithm, because most of that really is in the creation of the data set. Most of the details of how to use the profile kernel so Tobias Hemp did that, and essentially optimized the profile kernel for the subset localization prediction, and essentially used it the same way. And the only differences were the k-mers and essentially the way it's trained, and then he had to rewrite the program of the profile kernel, because for localization it sort of worked, but for protein actually he just had too much data, so the program was by far not fast enough. So he parallelized it, put it into a much faster, he sped it up by more than an order of magnitude. And there's a lot of work that was put into making this profile kernel work for the protein-protein interaction. And then it clearly outperformed other methods that were around. So this is for class C1. That's the new method here. And there are two new methods. I'll get to that in a minute. And for C3, the same thing. So for C3, in fact, not the same thing. For C3, it arguably does better than the existing methods. Even more, it does even more. Does even outperform the existing methods more. So here, it does not quite outperform the best method at low coverage and high accuracy. So you see that down here, this black method, which is pipe 2, is actually doing better than this profile kernel, but for C3 there's just no region where this doesn't do better. Now the two shades of blue are whether Tobias used expression and I believe, I'm not entirely sure whether I labeled that right, I believe, and subcellular localization. Now, the issue is, and that brings us back to Konstantin's your group's proposal of the hotspots. When you have the mechanism of a profile kernel, ultimately, remember the way it's done is you look for a k-mer that is conserved above some threshold in a family, all right? And then you put a set of these k-mers into an SVM, and it learns to essentially find which k-mers are indicative of protein-protein interactions, right? And this particular solution, the blue solution here, does this on using about so 50% of the information contained is contained in 10,000 k-mers, 50% in more than, between 10,000 and more than 100,000. So it's a lot of input. It's a huge input space that is used here. It's an SVM trained on more than 100,000 samples, okay? And they matter. 100,000 input dimension for very few examples. So we are clearly moving out of the comfort zone of 10 to 1. It's just in some sense the opposite. Not in some sense the opposite. But now we know that two proteins that are not in the same subcellular localization are less likely to bind than two proteins that are. Okay? Into the SVM profile kernel mechanism, formalism, we cannot put this information. It's not like a neural network where you put another input unit, and that input unit codes hotspots. That input unit codes same subcellular localization or not. No, the mechanism is essentially you have a vector product between things that are evolutionarily conserved or not. You could now introduce a new feature, evolutionarily conserved and localized or something like that. But then you suddenly blow up your input space tremendously. So one way to think about localization then would be you double it. So you have all the conserved motifs for same localization, not same localization. That's the minimal blow up, right? And that's an immense blow up if you go from 100,000 input units to 200,000 input units. So what else we can do is simply you say, okay, here's my prediction, and I will only take the predictions that are also predicted to be in the same subcellular localization, that are also predicted to be co-expressed. And that's the dark blue. Okay? That's the dark blue versus the light blue versus the dark blue. So the dark blue is I just take the profile kernel, the light blue is I take additional information, subcellular localization or expression. So this is labeled as expression, but I believe we use both here. And now, why should you do that? Because the blue is lower, the light blue is lower than the dark blue, right? But there's a realm where the light blue is better. And if we had more light blue, yeah, maybe it matters, right? So if that allows you to bring a discovery that otherwise is not discovered, it may matter, right? Maybe the dark blue is always biased by our way of doing cross-validation. The improvement over expression should be relatively independent because the expression data is there for everything. The more experimental data we get, the more the light blue should rise. But ultimately, when you see these curves, it's very clear what you put online is the dark blue. And that's in fact what we have done. So I'm going to slide through a couple of other methods that you can use to predict protein-protein interactions. The first one is the idea of duplication. So you have an organism P that upon becoming an organism, upon speciation, it can possibly include a new process. So when you now look at presence of proteins, so this is protein A, B, blah, 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 and this is one organism P and C. Now you simply ask, is this protein present in that organism? We build up a vector of presence. That protein is there, yes or no, okay? That's a cross. And when you go through all of these, now the in silico prediction essentially would be that things that somehow correlate. So clearly, if you do not have both proteins in both, or you have some correlation in the absence and presence, and that may get you to the point of in fact predicting an interaction. So, let's just keep it there. The idea is simple. It only works if you have very large profiles. So you need a lot of organisms, all the proteins that are common between a lot of proteins. And that's an idea that goes back to the group of David Eisenberg and Christopher Zunis. By the way, this, interestingly enough, in science is just one of those examples where these two methods here, essentially, I'm not sure whether you understood the idea that I convened, but maybe if I would be more clear, you would understand it. And then if you do understand it, then when you read these papers, you will see that both of them say that. But they completely did not talk. So they developed it independently of each other. Nobody copied the other one, and they published essentially at the same point. And the idea, how do these things happen, right? Is an idea that is somehow floating around. Very often people steal and copy. It's a common scientific progress, a means of scientific progress. This is an example where that didn't happen. Again, the two people, Christopher Zunis and David Eisenberg, is the main point behind that. The second idea is the idea of domain copy. So the domain copy idea is you have a protein A in organism X and a protein B in organism X. And now you have a protein C in organism Y, which has both domains. Okay? So this is a single domain protein, this is a single domain protein, and this is a two-domain protein. So then maybe the argument is organism Y has this as a single domain, as a two-domain protein, as a single gene, simply because these two things interact. And by putting it together on the genome, you increase the odds of making them meet, because they are already together. Okay, you solve one issue. So if you find these cases, then the argument is, then they are indicative of protein-protein interactions. You may not find them very often, but when you find them, they are indicative of protein interaction. Again, this is David Eisenberg. And again, so in this particular case, it's Terry Gesterland. And again, two groups had the same idea at the same moment, independently of each other. It's twice David Eisenberg, but it really... So I have a lot of evidence that I essentially know that, so they didn't copy. Trust me on that. And the third story I just discussed yesterday on the phone with David. And that is, no, it's the next one. So here's correlated mutations, the idea of correlated mutations. I briefly mentioned it in the last semester. So this is a family of proteins where the sequence of one protein runs in that direction. So these are all homologs. So this is the original sequence, and 1, 2, 3, 4, 5, 6, 7 homologs. And now assume you see a position where some set of the family have this molecule, the R, some the K, and some the W. And say there's another position where you see that exactly those three have some residue in common, exactly those three have another residue in common, and exactly those two have another residue in common. Then the suspicion is that what you have is a sort of compensatory change. That in fact, in this particular case here, it's not that easy to see, but so R and K are both positively charged residues of different sizes. D and E, for D and E the same is true. Now you imagine that they bind and that the R is a little bit larger than the K is a little bit larger than the R. This is a little bit smaller than this, so you essentially occupy the same volume of the contact. Okay, you have to flip in order to have a positive-negative contact here and bury the same volume. You have to flip it here from D to E, and vice versa. So essentially, in evolution, maybe this one is first. It moves from R to K by chance. And in order to not occupy more volume in the inside and sort of blow up the protein, you also have to then flip the D to E. So the best mutant that would be surviving here is one that goes from D to E. And then you have essentially maintained the binding of K to E or D to R. of positive to negative, and you have maintained the volume of the two. We have made one of the two a little bit larger, the other one a little bit smaller. The common volume is the same. And here you have created another way of making them interact, but in this particular case is not what is called a salt bridge. Whatever. What I'm trying to say is, if you see these cases, they are indicative that there is some correlated event. And this correlated event tells me something about likelihood of being close in space. Now, this likelihood of being close in space can now also be used to predict protein-protein interactions. So it can be used to predict internal residue interactions. In fact, that is the first time in history that the group of Debbie Marks and Chris Sander, I'm lacking here, and it was done by Thomas Hopf. And published in 2012 in his journal Cell. That was the first case where people, just from an amino acid sequence, could predict a three-dimensional structure of proteins that were not sequence similar to anything people had ever seen before. And not only that, on top they did it for membrane proteins that are very important for drug development. And those are three predictions that in that particular paper they put forward of proteins they predicted the three-dimensional structure for, but never anybody had seen. I'm not sure about those, but they put up 11 in their paper, 11 of these predictions, and 3 of those at least, or more than 3 have been verified by now experimentally. Anyway, so underneath this idea is this idea of correlated mutations. And this idea of correlated mutations you can also use to predict protein-protein interactions. It's slightly more complicated, but ultimately it's the same idea. You now assume that this is essentially, you cut here, this is protein A and this is protein B, and you see some correlated changes between protein A and B, and then you believe that they are correlated because A and B somehow evolve under similar constraints. And similar constraints may imply that they have to interact. Okay, that is the similar constraint. And the method here goes, however, back to something that Alfonso Valencia did initially. Now there are much better ways available, and that's the latest of Debbie Marks, where you can use that to really predict for some proteins very reliably protein-protein interactions. Now, the next idea is the one of a mirror tree. This is sort of more complicated. Ultimately, let's skip that. It's another idea from Alfonso Valencia. So this has to essentially have to do with the idea that you have two proteins. Let me just illustrate the idea. So you have two proteins or protein families, and you see that in these families you always have sort of the same species aligned. Okay? And then when you see that two proteins always have the same species aligned, then they are sort of common to the same set of species, unless all proteins in that organism are common to all species. That would be trivial. But it is actually not like that. Trees that you build for different proteins, human has 20,000 proteins. If you sort of look at the phylogenetic map of what they are similar to, most likely you get 10, I'm just making up a number here, but you get 5,000 different maps or something like that, right? And the few that are similar are either similar because they are similar sequences, or between pairs of proteins they're similar because they interact. And that's the idea about a mirror tree. And it somehow works. The next idea here is the motif idea. So that ultimately, underneath the idea is protein A and B interact because somehow there's a binding site that has a specific shape. And another pair of protein that has the same shape, not the same shape overall, but of the interaction site, which is roughly 10% of the protein, so it's relatively small, right? If I found the same sort of motif, then that would be indicative of them interacting. So what you would have to build then is a motif data set, a set of all the motifs of protein interactions. The challenge here is we don't have enough binding between proteins to define these motifs on a molecular level. Okay, so we have to define them on a sequence level, and then what you drag in is a lot of, let's call it dirt. And there's another case here, and this is Hanna Magalit, Andrew Ryszewski, where again this is not quite simultaneous, but the same idea was published twice independently. So this brings up a bunch of methods that work, and ultimately they all work for some cases. So they all don't cover, if I do all human all against all, they cover a tiny bit of it. When they work very well. But they are all not doing what the method that I showed you before, the profile kernel, is doing, which is essentially they perform for anything that we have never seen before. They are all sort of driven by some information that is already there. Here we have the idea of arrays. So you have data. I mean, here's the idea. Data that we can use in order to predict protein-protein interactions. So we have sequence similarity, we have phylogenetic profiles, we have domain interactions, we have possibly some idea about structure. There's this idea of gene fusion. So these are essentially the different domains that I talked about. The microarrays I didn't talk about. Microarrays now. So what you see is you have, for instance, you begin with cancer cells and non-cancer cells. You take the RNA out, you do some biological treatment, and then what you essentially get is a bunch of conditions. So now you see one gene, in this particular case here, is in this row, and the light or the color is indicative of how much of this protein is expressed. Okay, how much is expressed under particular conditions? So there's one condition, 2, 3, 4, 5, and so forth. Right? You see many conditions, and then you can immediately imagine that proteins that are somehow related will have a correlation of light. Okay, so if under the same conditions they're always red, and under other conditions they're always green, then you would assume that they are somehow associated. That they have something in common, because they react the same way, in some correlated way, to conditions. All right. So then the idea is take these conditions and see whether we can use that to predict protein-protein interactions. And I briefly mentioned that at the beginning of Tuesday, that you can use this by training a machine learning device, putting these conditions into it. Now, the next idea is use structure. So the structural motifs is one idea. So just drag out that there's this beta strand, or this pair of beta strands that are binding, and you can sort of generalize that and essentially find all possible structural motifs, and on all of these structural motifs build a machine learning device. And that is work that Barry Honig in Chang published. And here's one of those examples of, in their Nature paper from now 6 years ago now, one of the predictions that they had that was new. So nobody knew that this would bind. And overall what they say is that, so this is the random false positive rate, true positive rate. That's what their method does. And this is where experimental, according to their paper here, where experimental methods sit. So they argue that this is a method that may not, again, predict for all the pairs in human, but for some subset for which we have some structural information available, we can actually do better than some low resolution experiments. That's what this one says. Did I talk about the expression arrays?
I don't think so.
So when I said that this is what I talked about on Tuesday, I was lying. And nobody said that. That's work from Ta Tseng. Since I've been lying about it before, you already accepted that we talked about it. So you can do that. You can use, and again, let me repeat, in the context of the examen, if I fast forward through something like that, this will not be a subject of the exam. So there will be no question from this. But ultimately, at the end of the day, you can use microarray data to predict protein interactions, physical protein interactions, by, in fact, explicitly training a machine learning device. Now we get into the issue of database integration. So once you have one method, there's tons of these methods out there: the gene fusion, homology, microarray, functional similarity, mirror tree, maybe text mining, subcellular localization, domain, conserved, phylogeny, SVM-based protocol. So how do we combine them all?
With a naive base.
Yeah, do you know anything better? By the way, this is what I skipped on using microarrays was done by Tarzan Song, and Tarzan first did this naive Bayes, and then he sort of looked at the question: how much does a particular feature improve in this naive Bayes, right? So if I leave in this feature or leave out this feature, and then he saw that the co-expression, just like in the SVM profile kernel, the dark blue was above the light blue. In his case, the naive Bayes did much better if we left out the experimental information. And Tarzan felt this cannot be true. There must be information in there, so I must be able to specifically train a machine learning device to turn this around. So his PhD was supposed to sort of do exactly this and improve on the naive Bayes here. And the PhD turned into something else, where he established that you can use the expression array and not make it worse, which was a publication, which was interesting. But he never got much further into the integrative. We need to do something better here, and this is another research project that essentially this kind of naive Bayes is what everybody else does. I don't believe machine learning is maybe it is, but this is something we have to rethink. Now, that's the okay. So that gets us into the final part of the protein pairing part, and this is another one. Peanut, I will wrap up. Just the idea behind it is you have a bunch of proteins, you have some data about their function, you have some data about their interaction, and you have some data about their localization. And let's look at localization, for instance. You can compute a probability map that two proteins from the same localization interact. More than expected is green, less than expected is red. White means as expected, roughly, right? And on a scale of these three numbers, what you see is that mostly green is the diagonal. Most proteins that interact are in the same compartment, except for extracellular, but extracellular is sort of outside. And most proteins that are outside don't sort of interact with each other, but they interact with other cells, right? That's the reason why this one, or the rationalization why this one is white. Now, you can now turn this table as an observation, right? And you can take this observation and turn it into an annotation tool. You can use that in two ways. You can use subcellular localization to sort of draw your interactions differently. So you have a bunch of proteins that interact with this particular one here, and now you put them in different places of the cell because they belong to different subcellular localizations, right? You have what the field calls a hairball. So you have one protein interacting with many, many other proteins. And now you sort of drag the hairball out because some of those this one interacts with in this compartment, are in the nucleus, are in some vesicles, or are secreted. So you have this same information that you simply display in a different way. Okay? The other thing is that now using this table here, you can sort of compile a probability. So each of these annotations, each of these pairs of protein interaction but experimentally observed, you can say, well, I trust it according to this table. So if many of those interactions that are experimentally observed are in this red part, we say they are not very likely. Some of them may be right, some of them most likely will be right, but if many are, then there's something wrong. And that is something that you can put into the annotation. So you can essentially put, yeah?
And can you go one slide back? Why isn't it symmetrical?
Why is it not symmetrical? There's an answer. Let me get back to that. No, I have to do that now. No, I will not do that now. There's an answer to the question.
Yeah, okay, I believe that. So that's fine for me.
I have to think it through. In fact, the answer to this question prevented us from enforcing this. There was a reason why we did not enforce symmetry. Okay, now one particular example of this is a view of the Alzheimer disease pathway, where you see that the yellow one here interacts with many proteins, but only one of them goes through this set of vesicles. This one that goes through the vesicles has this name in the database. If you know what this means, it means that this is a protein for which there is no annotation of what it does. When you are looking at a yellow protein, and that yellow protein interacts with 150 proteins or more, and one of those is this one here, then this is the first one to ignore because there's nothing known about it, right? So what you as a user will do is you will follow up on all the others where there are some names, where there is something that you can look up and see whether it makes sense and whether you can learn something for your experiment. But this particular one you will ignore simply because it's a protein for which nothing is known. The moment we put it onto this subcellular localization map, you see that this is a unique path. Unique in the sense that there are many connections in different directions, but there's only one here. So this could be a mistake. True. But if it's not a mistake, then this is potentially very interesting because it's sort of a bottleneck, or it's a particular part of this pathway. And so there was some follow-up study done. So this is an older work. So this was an actually interesting discovery for some experimental follow-up. Now, Christian Dalago most recently tried to sort of implement the same idea on a different level in a cell map that wants to visualize protein-protein interactions and subcellular localization at the same time. And so this is the map of the cell. These are particular proteins. That is a probability derived from a variety of features, a probability vector. And so underneath now you can sort of put different maps into the system. The system has a bunch. The image doesn't come very well, I'm sorry. But you see some of these are fancy way of displaying, and then ultimately you define your map, or you use some of the maps that Christian is already providing. You use a set of interactions, or you provide your set of interactions, and that allows you, just like before, to go from the level of this hairball, where you have this one protein that interacts with many other proteins, and it's arguably not that easy to see a lot in this particular image here. It's not that easy in this one either. But it's a little bit better, is our argument. And we looked at than in any of these solutions that exist today or existed before. So he's publishing CellMap at the moment. I'm actually not sure what the status is, whether it's accepted or not. But our argument is not that we solve the problem of the hairball, but our argument is that even for a case that is as extreme as this one, we still create some advantage. We still create some points where you can see something, which is not at all the case here. The only way to go from this one here is to zoom into one particular one, focus on one particular one, and follow up on that particular pair. So randomly pick a pair. And the randomly pick a pair is just like before. You pick the ones that have annotations of names or something like that.